Hello everyone, my name is Dan Yi from the University of Arizona. Today my topic is endothelial SOX17 deficiency induces pulmonary arterial hypertension. Pulmonary arterial hypertension is characterized by the progressive increase of pulmonary vascular resistance and pulmonary vascular remodeling, leading to right heart failure and death. Genetic mutation and variants of SOX17 are associated with pulmonary arterial hypertension, but the role of SOX17 in the pathogenesis of PH is unknown. So we hypothesize that endothelial SOX17 deficiency induces pulmonary arterial hypertension. We used both in vivo and in vitro study, the RNA sequencing analysis and the transcriptional motif analysis to understand the molecular mechanism of SOX17 deficiency in endothelial cells. First, we found the SOX17 is decreased in pulmonary arterial hypertension patients' endothelial cells, both in messenger RNA and protein. Then we did an animal study. We used SOX17 endothelial knockout mice. We found the SOX17 knockout mice has a higher RVSP than the wild type, both in male and female, but there's no difference in RV-RV ratio. And the SOX17 knockout mice has a larger wall thickness than the wild type mice. For vessel less than 50 micrometer, SOX17 knockout mice has a higher vascularization than wild type, which is indicated by alpha smooth muscle actin. And this is the study of the endothelial cell. BRDO shows the proliferation, and the K-space 3-7 activity shows the apoptosis. We found the SOX17 deficiency can promote cell proliferation and decrease the cell apoptosis. The figure E shows the cell junction study, and SOX17 knockdown can impair the cell junction. The figure 4 is the RNA-seq data. A is a heat map showing endothelial SOX17 deficiency induced gene alteration. Figure B is the pathway enrichment analysis. We found the cell cycle is a main pathway. And the figure C shows this gene is the main gene in the cell cycle, has a higher expression in SOX17 knockdown group. And the figure D is a irregular motif prediction for altered genes in SOX17 deficiency endothelial cells. And we found the transcriptional factor E2F1. Figure E is a binding motif of E2F1. And the figure F shows the knockdown of SOX17 can increase E2F1 expression in endothelial cells. Then we use siRNA to knock down E2F1. We found the PLK1, CCNB1, CCNB2 can be increased by SOX17 knockdown and normalized by SOX17 and E2F1 double knockdown. BRDO shows the cell proliferation. SOX17 knockdown can increase the cell proliferation, and E2F1 SOX17 double knockdown can normalize the cell proliferation. K-space 3-7 indicates the cell apoptosis. SOX17 knockdown can decrease the cell apoptosis, and SOX17 E2F1 double knockdown can normalize the cell proliferation. So our study demonstrates that endothelial SOX17 deficiency induces spontaneous mild PAH. Loss of SOX17 promotes endothelial cell proliferation and anti-apoptosis, while the upregulation of transcriptional factor E2F1. Thank you.